simple act of breathing is actually much more complicated than it seems on the surface. This video is to elucidate us and make us understand as to what the factors are that encourage gas exchange inside our body. Our respiratory system works as both an inlet and outlet for a lot of byproducts that our body requires and or creates. Air enters our body through the nose, and in the nose there are present cilia, which are hair-like structures which help separate out big particulates from the air that we inhale. The nasal cavity also has mucosal membranes, which ensure that the air that we breathe in has a particular moisture content. The proximity of our respiratory system next to the circulatory system also ensures that the temperature of the air that we inhale reaches a particular point, which is very perfect for gas exchange. After the nasal cavity, the air passes through the pharynx, the larynx, the trachea, the bronchus, and the bronchi, and then finally into the alveoli. Alveoli are balloon like sacs which expand and contract as we breathe. Alveoli are what actually act as the gas exchange surface inside our body, and they do this by having particular properties which really enhance the rate at which gas exchange can take place. Their volume to surface area ratio is very particular. Their their structure is such that it is only one cell thick, so that the air that has to come in to our body and then go out of our body diffuses through only one single cell layer. And in addition to both of these things, our alveoli also create this thing called pulmonary surfactant or lung surfactant, which stabilizes the entire surface to ensure that our body keeps getting that constant supply of oxygen. The alveoli are covered by capillaries. These capillaries are also very thin. They are in fact so thin that only a single hemoglobin molecule can pass through them at a time. This ensures that gas exchange happens very well. The important factors when considering gas exchange are the difference in partial pressure, the total surface area, and the thickness of the membrane. These factors are automatically controlled in our respiratory system by this amazing thing called lung surfactant. The alveolar structure is made up of three types of cells. The alveolar cell type 1, which create the structure and give it rigidity and elasticity. Alveolar cell type 2, which create alveolar cell type 1, and the lung surfactant that we had previously mentioned. The third component is alveolar macrophages. Now, macrophages are cells that eat other cells. Similarly, alveolar macrophages eat those alveolar cells which have become dead or are decaying. In addition to this, they also take out any excess alveolar surfactant which has been secreted into the alveoli. As we breathe in, the alveoli expands and gets filled with air. This air is rich in oxygen and has very low concentrations of carbon dioxide and moisture. The capillaries, which are connected with the alveoli, are very deficit in oxygen and have high concentrations of carbon dioxide and moisture. Due to this difference in partial pressures of gases, gas exchange occurs and oxygen diffuses from the air to the bloodstream, while carbon dioxide diffuses from the bloodstream to the air. As we know, the alveoli are very delicate because they are only one cell thick to ensure gas exchange occurs. When we breathe in, the alveoli expands, but if we keep breathing in, will the alveoli just pop? What about when we exhale and the alveoli compress? Will the alveoli just sink into itself? The reason neither of these two things happen is because of lung surfactant or pulmonary surfactant. The pulmonary surfactant decreases the surface tension of the alveoli and increases its mechanical strength so that it can endure this process over and over and over again. The composition of lung surfactant is very complex. It is primarily made up of lipids and proteins, the majority of which being phosphatidylcholine in either its monosaturated form or disaturated form. In addition to these, there are also serine, glycerol, cholesterols, glycerides, etc. present in the lung surfactant. The proteins present also are very important. Special protein B and special protein C regulate surface activity and mechanical function, and special protein A and special protein D are acting as defensive agents against outside entities and pathogens. The interaction between lipids and proteins is what makes lung surfactants' properties very, very unique. As we have two phases of our breathing, inhalation and exhalation, the lung surfactant also has two orientations which support the inhalation phase and exhalation phase, as both require specific mechanical properties. This dyadic nature is ever present in the respiratory system. All of these together make a very complex monolayer spread across the alveoli. In addition to providing the alveoli with mechanical strength, the lung surfactant also acts as a defensive agent against particulates that enter our respiratory system. These particulates are usually in the size range from nanometers to micrometers, and they are a huge threat to the gas exchange process because they can alter the properties of the lung surfactant due to their high surface area to volume ratio. Even in the ideal case, when the particulate is non-reactive with the lung surfactant, even then the lung surfactant can be absorbed into the particle, or the particle can just provide steric hindrance, reducing the area of gas exchange. The alveolar cells, seeing this, can also secrete more or less of lung surfactant, which will change the gas exchange properties. Even if these particles reach our alveolar surface, all is not lost. Our bodies really know how to take care of themselves. Our lungs have self cleaning properties. Due to these self cleaning properties, all particles that enter our respiratory system are expelled. This occurs because of oscillatory nature of breathing and their anti-symmetrical elastic forces. These particles are expelled out from our alveoli and then our lungs because of phlegm